I went to Caltech, and I became interested in immunology just before I went to Caltech. There was a guy called Dick Dutton, who's also one of the past presidents of AA, who gave what now I realize is a job seminar at UCSD. I went to his talk, and I had always been interested in development, and I thought with incredible naivete that the immune system would be easier to understand and be the perfect model system for development. And when he gave this talk, there were only two kinds of immune cells, the ones that stuck to plastic and the ones that didn't, and he showed they had to interact to make an immune response. And I said, well, this is way cool. And so that's what I decided I wanted to do. And when I went to Caltech, I had originally thought I was going to do developmental biology and was going to work with a guy called Giuseppe Attardi. And after I got interested in immunology, I walked into Ray Owen's office and said, with amazing chutzpah, that I was going to work in his lab and I was going to be his graduate student. And I guess he just was so taken aback, he said yes. So I started working for Ray, and it was great. So he was the perfect mentor for me. He never ever told anybody what to do, and his guidance was modest in that I got my thesis project. He said, Jeff, I think transferins are interesting, and pigeons are kind of interesting, and Sandy Webb, who was a technician in the lab, knows how to run starch gels, and so you can actually, there are multiple alleles, and she'll show you how to do that, and I think something interesting is there. So that was my thesis project, right? So that was kind of non-guidance. But in fact, it was wonderful. So I went, and Sandy taught me how to run starch gels, and we started futzing. It turns out transferrin is found in egg whites. It's the second most abundant protein in egg whites after albumin. And in the body, most of the time, transferrin functions as an iron transport protein. It takes iron away from iron stores and delivers it to reticulocytes to get incorporated into hemoglobin. But in the egg whites, there's no iron in the transferrin, and it's bacteriostatic. And so it's part of the innate immune system in a way because it keeps the egg sterile. Lots of bacteria have a trick. They know how to go through egg whites. And I don't know how much you know about bird anatomy, but after you talk to Max, probably a lot, because birds have a cloaca, which is their all-purpose vent at the back end. So the genitals and the intestine all go through the same place, which you can imagine is dirty. So there's lots of bacteria, and so eggs have to have a way to stay sterile. If eggs don't stay sterile, then the embryos die. So transferrin keeps the egg sterile and allows the embryo to develop, and also the transferrin also gets absorbed by the developing embryo and gets used as its own transferrin. So as a graduate student, my very first paper was a science paper where we showed that the source of transferrin in a newborn chick comes from the egg white. It's not made by the chick itself. And we did that by using genetically marked transferrin. We also went on to show that females who are heterozygous for these two alleles at the transferrin locus are more fertile, and that the fertility, at least in part, can be ascribed to the fact that the heterozygous, that is having both kinds of transferrin present, are more bacteriostatic than having only one kind. And then I finally went on, and this is one of the great things where I learned that sometimes it's really good to collaborate. We tried to make a mathematical model for how to do this, for how that kind of fertility, which is not the sort of standard overdominance for fitness that you think about for resistance to malaria by sickle cell anemia, because it's kind of a twist on a fertility model. And I tried really hard for about 3 weeks to figure out how to make an algebraic model to do this. And Ray said, you know, I know this guy Jim Crow in Madison. You should just go over to see Jim, and he'll help you figure this out. And so we bought a plane ticket. I flew to Madison. I stayed at Jim's house, and Jim just died recently. But and in about 3 days, really 1 hour of his time, 3 days of my time, we figured this out. We published a paper full of equations in the American Naturalist. And it showed me that the right person can really fix a problem, right? And you don't have to know how to do everything that you need to do. You just have to know how to get help to do it. And I don't know if you know about Jim Crow. He's a really famous population geneticist, but just the nicest man, and I think he played the viola. Maybe it was the violin, but